Alright, so I'm gonna talk about the restriction endonucleases. And what this is with has to do with bacteria and how basically bacteria's enemies are called bacteriophages, which is a virus that goes into the bacteria and multiplies until the bacteria eventually bursts and produces more viruses. Well, now there's some bacteria that are developing through natural selection, and they have the restriction endonucleases, which is able to read sequences of 4 to 6 nucleotides that are usually palindromes. And through this, they are able to they go through a recognition sequence, which recognizes 4 to 6 nucleotides. And what this does is it makes like fragments of the DNA or the DNA duplex, and it just makes fragments of them, and the bacteria is protected from it because of the what's it called? Oh, the bacteria is protected from an enzyme called methylases, which adds methyl groups to some nucleotides on the bacterial DNA recognition sequence, so that the endonuclease can't bind to it. And all right, so to start with the process here, you have the DNA duplex here, and then with the endonuclease, the restriction endonuclease, there's a cleavage, and it basically creates fragments from the DNA we have here. And here we could see there's 3 fragments, and they're complementary. And first off, there you can see that they're palindromes. Here we had GAATTC. At the bottom, it's just the opposite. Same goes over here. Okay, so then we have this cleavage, and then it gets cut. These ends here that could bind together eventually are called sticky ends. And from here, we have if fragments are created from the same endonuclease, they can eventually bind together. And that's what we have here. You have this fragment over here, G, binding to this fragment on this side. And then you have this fragment down here binding with the one over here on this side. And basically you have the recombination DNA molecule. And in order for this to occur, DNA ligase helps attach them together. Next, I'm gonna explain how you could use restriction endonucleases to manipulate genes. And basically right here, we have PSC 101. Basically, this was using an endonuclease called EcoRI, and it cut a plasmid into fragments, and one of them was around 9,000 nucleotides in length, and it had the origin for replication to replicate the plasmid, and a gene that was resistant to the antibiotic tetracycline. And just because both these fragments were made by the same or cut by the same restriction endonuclease, they could be formed together, and this could create a circle that we see here. And after this occurred, we have here they used amphibian DNA, a frog, and they got the RNA using an endonuclease, the same one from before, the EcoRI. And this made a fragment of RNA, and they mixed it together with the plasmid that they had from earlier. And together they had formed a recombinant plasmid, and from this they had bacterial cells which were immune to tetracycline, and they also began growing frog RNA, which is a recombinant DNA. And this was surprising at the time because they discovered that they could create or manipulate genes that they never knew before.